Welcome children in bio class. Children, now I am going to teach you about genetic engineering. Manipulation of DNA to change its structure is called genetic engineering. Genetic engineering is a popular term used for recombinant DNA technology. Recombinant DNA technology involves combining of DNA from two different organisms and to generate a recombinant DNA. So their DNA of two different organisms are going to combine. Children, recombinant DNA is the hybrid DNA from two different sources formed by the union of foreign DNA and vector DNA. Foreign DNA may be from bacteria or from eukaryotic cell. It is constructed artificially in the laboratory. Recombinant DNA is also called heterologous DNA or heteroduplex DNA. Recombinant DNA technology is the usage of technology to alter the genome of a living cell for medical or industrial use. This is also called gene manipulation. Children, you know genetic code is similar for all living beings. DNA segment with structural gene and their promoter can be taken from one organism and incorporated into another organism. It may be taken from man and incorporated into bacterial cell. It includes the DNA segment or gene to be introduced. It is obtained by artificial synthesis of gene in vitro or formation of complementary DNA from messenger RNA by reverse transcriptase or isolation of desired gene or desired DNA segment from the original source. Isolation. Next step is introduction of desired DNA in vector DNA for obtaining recombinant DNA and its clone. Expression of recombinant DNA in culture medium. Use of clone DNA for the modification of genome or for the addition of functional gene or genes at desired point or desired organisms. Repair of defective cistron or DNA segment by using genetically engineered DNA to change defective phenotype. Combining genes from different organisms or the synthesis of recombinant DNA to obtain genetically modified organisms. Children, here you can see there is a bacterial cell and cell containing gene of interest that we can say desired genes. Both genes are coming together and then recombinant DNA plasmid is formed. Now this plasmid is put into the bacterial cell. Bacterial cell is the host cell. It grows in culture. A clone of the cell containing the cloned gene of interest. Now gene of interest are produced and it is producing the protein. So from this we can get the copies of gene and we can get protein also. So this is the diagrammatic representation of basic steps of recombinant DNA technology. So what are the basic steps of the recombinant DNA technology? In that, first one is the DNA segment or gene to be introduced, introduction of desired DNA in vector DNA, then expression of recombinant DNA in culture medium, use of clone DNA for the modification of genome, repair of defective cistron or DNA segment, and last is combine genes from different organisms or synthesis of recombinant DNA. So children, I told about the recombinant DNA technology. There in this I told how this in the recombinant DNA technology, how we are getting the hybrid DNA from the two different sources. That is, there is union of foreign DNA and vector DNA. So in this there is combination of DNA from two different organisms to generate a recombinant DNA, and it is also known as rDNA, and this recombinant DNA technology is known as genetic engineering.